Hey Mustangs, in this video we're going to be taking a look at organelles that you find in eukaryote cells. Now, when we talk about these organelles, we're going to be looking at their functions, so what their job is, what they do within the eukaryote cell, and also looking at what they look like as well, because you have to have an idea of what they look like, their overall shape and structure. All right, first thing that you really need to know and understand before we go any further is prokaryotes and eukaryotes. If you took all the cells that we know of on our planet, you'd be able to divide those cells into two groups, the prokaryote group and the eukaryote group. The prokaryote group has very unique structures and functions that are a part of it, and eukaryote cells have their own unique structures that are a part of them. So when you look at prokaryotes, prokaryotes are cells that do not have a nucleus. So if you looked inside of a prokaryote cell, you would not find a nucleus. They have no membrane-bound organelles. I'm going to talk a little bit in more detail about that in a little bit. They are single-celled organisms, so they're single cells that live by themselves usually. And the example we know of on our planet is going to be bacteria. So bacteria are examples of prokaryote cells. Eukaryotes, on the other hand, this other group of cells here, are cells that have a nucleus. They do have membrane-bound organelles. They can be single-celled organisms, but usually when you look at eukaryotes, you're going to find that eukaryotes are multicellular. So they're organisms like us, where there are multiple cells all working together as one. And the examples that we know of, the main examples that we know of are plants and animal cells. So plant cells and animal cells. Now, in order to help remember this, remember the saying pro no you do. So pro no you do means prokaryotes have no nucleus and no membrane-bound organelles, while eukaryotes do. So it's a little saying that a student came up with about 10 years ago that I had in my class, and I've been using that ever since to help students remember that prokaryotes do not have a nucleus or membrane-bound organelles, but eukaryotes do. So always remember that saying, pro no you do. Now, these membrane-bound organelles, it doesn't mean cell membrane. So we're not talking about the membrane that surrounds the entire cell, because even prokaryotes have a cell membrane too. So prokaryotes have a cell membrane, eukaryotes have a cell membrane. When we talk about membrane-bound organelles, these are organelles that have their own personal membrane. So within the cell, they themselves have their own personal membranes that surround them. Most of them have a single membrane that surrounds them. Some of them have multiple membranes, like the mitochondria and chloroplasts. They have actually two membranes that make them up. So the main membrane-bound organelles are the nucleus, Golgi, mitochondria, chloroplasts, rough ER, smooth ER, lysosomes, and vacuoles. Those are all membrane-bound organelles. Now, when you look at their membranes, they're very similar to what we see here in this plasma membrane or cell membrane that we see here, the membrane that surrounds the outside. So if you were to zoom in on their membranes, you'd see that they have a lipid bilayer. They have proteins that are embedded into the membrane as well as carbohydrate parts and stuff like that. Now, you wouldn't see the extracellular matrix and stuff like that because this is a cell membrane we're looking at, but the membranes of organelles would be very similar. So they have that lipid bilayer, proteins, carbohydrates that are part of them. All right, so the first organelle we're going to take a look at is the nucleus. Nucleus is the boss of the cell. So the nucleus contains the DNA and it's going to control cell activities. That's the job of the nucleus. The nucleus has these little circles, these little holes that we call nuclear pores, and these nuclear pores allow stuff to go in and out of the nucleus. And then inside, if you look at the DNA, the nucleolus is a kind of a condensing center there that we see of the DNA, and you see chromosomes here. So your nucleus is the organelle that contains the DNA, stores the DNA, and is the control center of the cell. Ribosomes are where proteins are made. So very, very important organelle, because we know that DNA is a set of instructions for making proteins, and then those proteins build and maintain a living thing. So ribosomes are the actual organelle where proteins are built. So if we take a look at ribosomes, they're usually represented as little dots in a drawing of a cell. Some are attached to the rough ER here, others are free-floating ribosomes, but in any case, ribosomes are like mini little factories. So many little factories that are going to build the proteins by connecting amino acids together. And if you think about what a factory is a place where you assemble a whole bunch of parts, and that's what's happening here. You're assembling a whole bunch of amino acids in order to build a protein. And then here is an actual cellular representation. All this, what you see here, these little foldings, that's the rough ER, but all these little dots on it, those are the ribosomes. All right, then you have the rough endoplasmic reticulum. Rough endoplasmic reticulum is the organelle right outside of the nucleus. Sometimes people confuse the rough ER with the Golgi apparatus. The rough ER, like I said, is right outside the nucleus, whereas the Golgi is usually off way over here. So it's not right next to the nucleus like we see with the rough ER. So the rough ER is kind of like the cell's highway. So it's a transport system that allows stuff to move through the cell, and here is the actual microscope picture of the rough ER. And you can tell the rough ER from the smooth ER because the rough ER has ribosomes attached to it, but the smooth ER does not. So the rough ER has the ribosomes attached, and so when proteins are actually made in the ribosomes, the rough ER is actually going to be used to transport the proteins through the cell. All right, then you have the Golgi apparatus or the Golgi body. So it's this membrane folded structure here. So here we see an illustration and then an actual microscope picture of it. The Golgi apparatus is going to sort, package, and secrete proteins. So it's often known as the post office of the cell. So if you think about what a post office does, it collects mail and then it sorts it out to where it needs to go, and then it finally sends it to where it needs to go. So that is what the Golgi apparatus is going to do. It's going to sort out proteins, package them, and then send them where they need to go. In order to send these proteins through the cell, and when we say it packages them into vesicles. So remember, vesicles are little structures of a membrane that surround the proteins in this case and transport them through the cell. So if we look at these organelles that we just went over, the nucleus, the ribosomes, the rough ER, and the Golgi apparatus, they're all involved with proteins. So the nucleus has the DNA that has instructions for building proteins. The ribosomes, then instructions, you make a copy of instructions, that copy gets sent to the ribosomes. The ribosomes make the proteins. The rough ER is going to send them to the Golgi. The Golgi will then sort them out, package them, and send them out in a vesicle. So you can see a vesicle. Vesicles actually bud off. So a vesicle buds off from the rough ER to go to the Golgi apparatus. And then here, the Golgi apparatus will package the proteins into vesicles and send them where they need to go. Some proteins stay within the cell. Other proteins actually leave the cell. All right, the mitochondria. Mitochondria is often referred to as the powerhouse of the cell. The mitochondria is the place where you transform the energy from your food, stored in lipids, proteins, carbohydrates especially, into ATP energy. So that's why it's known as the powerhouse. Now, the thing that's unique about the mitochondria is the fact that it's one of those organelles that has two membranes. And another interesting thing, it contains its own DNA. Okay, so that's what's interesting about the mitochondria. The chloroplast is often known as the solar panels. So it's the solar panels of the cell, and its job is to capture energy from the sun and convert it. So if you think about what solar panels do, solar panels are going to trap energy from the sun and then convert it into electricity. Here, energy from the sun is trapped and then converted into carbohydrates in this case. So remember G3P, which can then be made into glucose. Unique thing about chloroplasts is the fact that it has two membranes, and it also contains its own DNA. And both of these, the mitochondria and the chloroplast, are part of the endosymbiote hypothesis, the idea that a larger cell engulfed smaller prokaryote cells, and they became symbiotes. They basically developed a symbiotic relationship where the mitochondria or the chloroplast lived inside of the cell and made stuff for the cell, and the cell provided like a safe place, and basically they work together. Then you have the smoothie R. Smoothie R, remember, has no ribosomes on it. The thing I always used to remember the smoothie R is smooth like butter. Butter is a lipid, and the smoothie R makes lipids. Lysosomes. Lysosomes are organelles that are there to break down and recycle waste within the cell, also viruses, old organelles. So it's often thought of as the recycling center of the cell. Inside of lysosomes, they contain enzymes that are going to be used to break things down. So without those enzymes functioning, the lysosomes can't do their job. So enzymes are a very, very, very important component of the lysosomes. They are the things that break stuff down within the lysosomes. Then you have vacuoles. Vacuoles are usually used as a storage center for pigments, other materials, and in some cases they can even be used to digest cellular materials, very similar to the lysosomes. Last, we have the centrioles. Centrioles are organelles that are used to move chromosomes around during mitosis and meiosis. And this is a very important job, very underrated organelle. So they move chromosomes around during mitosis, meiosis to get them in the right position, make sure copies get pulled apart, make sure homologous pairs separate due to the law of segregation. So they are, you kind of think of them as a movie director that tells things where to go. Here, the centrioles are moving the chromosomes around to where they need to go. Oh, there's one more, my bad. Cytoskeleton. Cytoskeleton is, think of the frame of a house. In order to build a house, you always see them put up the wooden frame first, and then they build the house around that. It's the support of the house. Here, the cytoskeleton is provides shape and support of the cell. In order for the cell to remain and retain its shape, the cytoskeleton is what's used to do that. If you took the cytoskeleton out, the cell would actually collapse. All right, so when you do your cell brochure, you're going to make sure that you pick a theme first. So if you pick a theme, one really easy one is like an amusement park, and then you take that, and every part of the amusement park could be connected to one of the organelles we just discussed. So you're going to do all those organelles, plus you're going to want to do the cell membrane as well. So, for example, if you're doing an amusement park, really easy one to do, the trash cans and the recycling cans are like the lysosomes, because it's a place for stuff to be deposited and then it'll later be broken down, whether it's recycled in recycling bins or taken to the dump where it's going to get broken down. So the trash cans are like the lysosomes. Some people say the restaurants are like the, not ribosomes, lysosomes. The restaurants are like the ribosomes because that's where proteins are made that people are going to eat. So you could say something like that. And then you have, I messed up. You have the mailbox. This should say Golgi Apparatus. So the mailbox here, you could actually deposit letters, like Disneyland has its actual own post office, and you could deposit letters and then transport. They'll be separated and then transported to where you want to send them. Now you're going to want to make it like an actual brochure, so you could take a piece of paper and fold it and separate things out and kind of make it look nice. Doesn't have to be really, really good. I'm not looking for that. I want you to just do this so that way you can remember what each organelle does, to get their functions stuck in your head by connecting it to a theme somehow.